So I really like the story of the first artificial pea being created back in 1828. Why is that so important? That they were able to make pea, something that you can do pretty easily without all that fancy equipment. Well, the main thing is vitalism was the main belief that people had back to Aristotle up till the 19th century that some organic compounds things from living created by plants and animals, fungi, and of course bacteria, archaea, require some sort of mystical magic force. Vital is kind of what it came to be known as, and it's kind of a religious connotation, things we can't know about. And humans, a lot of times we haven't been able to understand things, so we kind of say it's kind of a mystical thing. But by taking raw, starting, basic chemicals, they were able to make pee in the lab, and then they were able to say, hey, these complex organic molecules are governed by all the physical and chemical properties that we're now beginning to understand more, and we can explain it. So that kind of got organic chemistry started. It's kind of like the birth of organic chemistry. Really started being able to manufacture these molecules. So basically, you need to know all the basic macromolecules, carbohydrates like glucose, amino acids, and fatty acids, triglycerides. And you need to be able to recognize the structure because on the exam, they will throw these molecules, these molecular diagrams out. So, best way to learn these is to practice drawing them. So you can see me, I practice drawing this. I got my glucose showing all the atoms and bonds, and you'll see glucose is 6 sides. Inside the ring, you'll see this oxygen, and then connected off to the side, you'll see the CH group. And I guess if I really draw it out, carbon, hydrogen is there, O, and then an H. That's kind of this. Explain it a little bit better, coming off the carbon from right here. You'll notice when I draw this, where there's this junction, I don't draw a carbon because that's kind of what you basically do. You draw the ring and you just assume that's a carbon there. But let me go back and put that in there so you know that's a carbon. 2, 3, 4, 5, and then 6. That used to kind of confuse me because I'd see the ring and I'd see, oh, there's 5 carbons and oxygen. I would forget that there's that carbon off to the side there. So practice drawing that. Draw more than once. Repetition is important when you're learning stuff. And then you'll move on to ribose, and you'll see this is a 5-ring sugar. Instead of hexagon, pentagon, you have 1, 2, 3, 4, 5 carbons involved. And if you get rid of this hydroxide, just have a hydrogen, then you get deoxyribose, which you know is DNA. So this right here is what you would find in RNA. Let me switch to a more visible color. So this RNA. So definitely go through and highlight everything. Carbon, carbon, not highlight, draw it. Make sure you draw this carbon. So 1, 2, 3, 4, 5. Remember, this is what's in RNA. Remember, you got your DNA in your nucleus. It gets converted to RNA, which gets converted to a protein. Basic central idea of how organisms use their DNA, but a lot of this RNA doesn't get used at all for protein. It goes back and modifies the DNA or modifies the making of other proteins. Pretty complicated. This is all you need to know right now. Plenty of time for complication later. Trust me on that. So you've all heard of saturated and unsaturated fats. Undoubtedly, we're very fat obsessed in this country. And let me tell you what we're talking about. You'll see, if you just look at these, you'll see, should notice you have a double bond here. Double bond. and that's what makes it unsaturated. See how you're missing hydrogens? So our hydrogens aren't there. That means it's unsaturated, means something is totally full. So totally full of hydrogens. Completely bonded out with all the carbons. Here, we're not totally full. So unsaturated, not full of hydrogen. So you'll want to draw that as well. And you can see this is saturated, so I don't have any double bonds in there, all single bonds. But if I wanted to throw in a double bond, that would take away two of my hydrogens. I'd be missing one here, and I'd be missing one here. So it's unsaturated. And amino acids are other major macromolecule. You can see the basic. This is the amino group. Has the nitrogen. This is, you guessed it, the R group. This right here is called the carboxyl group. And this is our central carbon, and you have this hydrogen. And just take a note, for this R group, there are 20 types of them. So there are 20 different configurations. Some is just an H, some there's a ring shaped molecule there, some you have a more highly branched molecule. We'll get into that later. Remember, plenty of time for complication later. And then you should draw this, remember, repeating to yourself, always clarifying carboxyl group, amino group, and then your R group. And you might have heard these words catabolism before. And anabolism, when I first hear of it, you're talking about anabolic, or I've heard anabolic steroids. And that's how I would remember if you've heard anabolic before. You know, you're using anabolic steroids, people are building up muscle, so you're building. And you're making stuff, basically. You're making stuff. In catabolism, you're breaking stuff down. So like you're burning stuff for energy, you're digesting stuff, you're getting that energy back, okay? Anabolism, a great way to think of it, we got photosynthesis. And you're making the sugar, right? But then you want to eat it, you got to break it down in respiration. That's catabolism, respiration. And you see that little chart I put there for you. And here's another explanation of it. Remember photosynthesis, anabolic, and I think I have behind here burning energy. This is respiration. Is catabolic.